Yeah.
Oh.
So yeah, but I only.
Yeah. No worries. Yeah, just get into the Zoom room. Tell Ricardo to give you guys permission to share. Can you give Matt and Megan permission? Yeah, and then you should just be able to share from your laptop. Okay, we're gonna be on the same. You just joined, so I just promote you.
Yeah.
Yep.
Yeah.
Okay, we can go ahead and get started. Thanks for coming out, everyone. This is kind of a cool opportunity to learn what the postdocs and research associates in the department are doing. And just, fun talks and science, and we'll have a reception after. We have 7 people, so I want to make this quick. You can go ahead, Sarah. So first up, we have Sarah.
All right. Hi, everyone. Nice to see you. For those of you I don't know, my name is Sarah Gravem. I started as a postdoc in 2015, and I'm a research associate now. I work really closely with Bruce and the PSCO group. But I'm actually not going to talk about any things that I do with Bruce today, so that's kind of fun. I'm going to talk about a collaboration I have with people at UC Santa Cruz and U of O, looking at sunflower sea stars. So. Okay, oh, weird. Oh, well, that's how it's gonna look. I don't know why looks awful, but we're gonna go with it. Okay, so I'll be talking about a project called Star Trek, the Landscape of Fear. And sorry, I can't see anything because of Zoom controls. The Landscape of Fear created by sunflower sea stars hunting in an urchin barren. And I just wanted to recognize the many, many students, including OSU students, but Santa Cruz and UO students who've all contributed substantially to this work. And none of that's working. Okay.
You need to click the presentation.
Okay, there we go.
Zoom.
Pardon?
You need to click the presentation after clicking anything on the Zoom background. Otherwise, it doesn't advance.
Thank you. Okay, so for those of you who don't know, this is a sunflower sea star, and it is Pycnopodia helianthoides. It gets about this big. It's like an extra large pizza. It has over 20 arms. It is a ravenous predator on the seafloor. It is super fast. It's one of the fastest sea stars in the world, which isn't all that fast, but you can see it cooking on the seafloor. And unfortunately, it was hit really hard by the sea star wasting disease epidemic that happened in 2013-14. So it's been a decade now since this happened. And this sea star was the hardest hit. So Sarah Hamilton and I, who was a graduate student in Bruce's lab, did a global synthesis of the sunflower sea star. And it was for the IUCN Red List. And we found that as a population, they declined by over 90% worldwide. Over 5.75 billion animals died with a B, which is bananas. It's the largest marine epidemic ever. That qualified them for critically endangered on the IUCN Red Listing rubric. And the devastation was regional. So here in Oregon, we lost over 98% of our sea stars. I'd say that's actually 100, but temporarily we lost 100. And that was the case all along the contiguous outer coast of the US, all the way from outer Washington to Mexico. So it was like essentially gone from that entire section of its range, which was about a third of its range, which was nuts. And then north of that in the Salish Sea, they held on in pockets, and that whole section up into the Gulf of Alaska was over around 90% inclines. So that is now being considered as an endangered species by this Endangered Species Act, which I helped review, and that should be coming out at the end of summer. It'll probably be listed as threatened, not endangered. So this is important not only for the sea star, but because it is a really key part of subtidal ecology. So they're a sea urchin predator. That's a little purple sea urchin that's having a bad day underneath that Pycnopodia. And this matters because sea urchins eat kelp, and kelp is the base of the subtidal ecosystem, at least on our coastline. So this is an urchin barren that's expanding to the edge of a kelp bed and taking it over and doing its little urchin front thing. And this was something that coincided with lots. So the Pycnopodia die-off happened first, right? In about 2013 or to 2015, depending on where you were. We also had a really huge recruitment of sea urchins right after that. And we think they were related. No one's actually certain whether that recruitment event was related, but it's very curious. And then a marine heat wave came by. It was called the blob, which killed kelp directly in 2015 and 16. So the sea urchins, sea stars went down, the sea urchins went bananas, climate change came in, and it's just perfect storm for kelp. And so ever since then, the sea urchins have been increasing all over from Central California to Alaska, and the kelp has been declining to the point where like it's called collapsed in much of Northern California and Oregon. So everybody knows about trophic cascades, right? So in this case, sea stars eat sea urchins, eat kelp. And that is a very common way for us to think about how ecosystems work. And everybody's looking at me going, where's the otter? Because everybody loves otters because they're cute and they're cuddly and no one's actually ever met one. But no, it's not true. They're pretty mean. And but if you think about it, we haven't had otters on our coast for 100 years, right? But we've had kelp all that time, presumably doing well. So we had otters for 100 years, and then within the decade after sea stars went kaput, the kelp collapsed. So we think that these things are related. And as much as otters are probably very, very important for kelp forests when they're there, we're interested in how important sea stars are for when otters aren't there, right? And the other thing to note is even if we had otters on this coastline right now, how many do you think we'd have? Like a couple thousand maybe could handle, this coastline could support that. We used to have 70 million sunflower sea stars. And so even if an otter can eat 10 urchins a day, a sea star can eat 2 urchins a week. But you multiply that by 1,000 otters versus 70 million sea stars, and you can start to see how they might actually play a big role as well, right? So we're really interested in not only how many urchins these sunflower sea stars can eat or used to eat, but also how they scare the living spines off of these little suckers, because I'm going to show you a video that was taken in an urchin barren in Fort Bragg. This is the first sunflower sea star they saw in California in over like 6 years, like the first confirmed sighting. And this urchin diver got a video of it. And so what you can see is there are no kelp on the seafloor. It is thousands and thousands of urchins. And as we pan over, I want you to see if you can find the pycno. Okay, so we can see it, and the urchins are running up this wall, right? We call this an urchin stampede. And what you can see is this guy's fat. So he or she probably has a couple in its belly already, and it is going for more. It's hunting up that wall. It's trying to grab those couple. Those couple are snugged up against the others and they can't get over the top of them. And I want you to, as we reverse out of this video, see if you can tell where the sea star has been in the last few hours or days. So do you see that big bare patch? So you can see its wake as it like strolls its way and slimes its way through this ecosystem. So not only can it eat a couple urchins a week, it can scare thousands of urchins an hour maybe, and potentially change their behavior for hours or days or even months if you multiply that one guy's activities by 70 million, right? So we're really interested not only in the trophic cascade part of it, but the behaviorally mediated trophic cascade. So how it changes the urchin behavior and changes the kelp forest. So this work is with Christy Croker from UC Santa Cruz and Aaron Galloway. And we have a lot of different projects that all interlock on this theme of trophic interactions and behavior. I'll talk about a couple of them today. but know that there are more. And so the first one I wanted to just highlight really quick was by Miles Rao. He's one of our undergrads. He was a SURE student in my lab last year, and he went to Sitka for the whole summer and dove almost every day. And he was asking whether wild pycnopodia reduce prey densities locally. So essentially mimicking that video that you just saw and putting data to that. So we're up in Sitka because you literally can't find kelp, urchins, and pycnos in the same place anywhere else that is easily accessible by boat, unless you go to Canada, which is hard. And if you're in Washington, it's a lot harder to get all three in the same place. And here we don't have any pycnos. So we had to go all the way to Alaska to do this. And what he did was he'd go out on a dive, and when he found a wild pycno living its life, he'd slap a quadrat down on top of it and then do radiating quadrats out from the middle of that pycno in all four directions, and then quantify how many grazers he saw in each quadrat. So abalone, green urchins, and red urchins. You'll notice purple urchins aren't on here because there aren't any purple urchins in Sitka. Well, there's like 7. And then from the pycno, this is like right on top of it, and that's 4 meters away. So we're looking for slopes in that line, right? And so what you can see is that the abalone are reduced slightly closer to the pycno, but there's more of them further away. And then same with green urchins, they're reduced closer to the pycno, and there's more of them further away. And we were like, duh. And then we got to the reds, and we were like, what the heck? What are the reds doing? Why are they not scared? And then we were like, I wonder how many red urchins are at that site normally. Like, what is the background density? And so we took and did a survey of background densities at these sites, like a well away from a pycno, and mapped that onto the graph. And what you can see for abalone is around 4+ meters, they reach their background density. So like where those two lines intersect. And same with greens, around 4+ meters, they start to reach their background density, but reds never reach their background density. So we think that the 4-meter halo of that pycno being around is actually bigger. It's actually more like 6 or 8 meters. So that's one thing we're going to do this summer when he goes back. The other thing that he found that I'm not going to show you is that when the stars are active versus inactive, these relationships get stronger. But he only found 4 active ones, so he's got to go find more active ones. All right, so the next thing we did was, so we've got how do they affect local densities when we stumble upon them? What would we do if we actually reintroduced them into an urchin barren, which is what essentially we're trying to figure out whether it's worth spending millions and millions of dollars to do here. So we did star track to put these pycnos on a hunt and find out how grazer behavior was affected. So we would release them on the seafloor. We're tracking where they go using these little flags and videotaping using a big tripod underwater. And what we'll do beforehand is capture the starting grazer density. So we say, okay, this is where we're going to put the pycno, and we do radiating quadrats outside of that and get grazer densities. And we also had control tracks so we could see what happens in a paired non-pycno track. We release the guy or girl and let it hunt and see what everybody does, and then resurvey in the track and then radiating off the track again an hour later, and then the next day, and then the next day, and then the next day, to see if not only do they flee, but do they stay away or do they come back, right? So this is what it looks like when you slap a pycno on the seafloor. So those flags go down every 2 minutes. The camera moves when the pycno moves, and we're tracking it for about 20 minutes. And you can see all these little green urchins, all these little abalone, and all these red urchins freaking out and running around, and they're scared. So what does this look like in the data? So we again sort of have this zero line, which is this is a line for no difference in grazer density between the controls and the C-star additions. So the Y is relative grazer count. So if it's below 0, the grazer count went up compared to the beginning. And if it's, if it's above 0, went up compared to the beginning. And if it's below 0, it went down compared to the beginning. And what we find is that nearer to the Picno versus far away from the Picno, you see that there's a drop, right? For all of abalone greens and reds. Tegula do, I don't know what they do. They do crazy things. They run around in circles. They're only like this big. So I think they just don't have a really good sense of like where the predator is in relation to them. They can also make it through a Pycnopodia's gut for it can be in there for 2 days and they spit it back out and then the Tegula just walks away. It's amazing. All right, so the abalone are scared. They run away, right? And same with the greens and the reds, but we want to know how far away, right? So we're trying to figure out where that line intersects with the no effect line. And for abalone, it's around 3 meters. And for greens, it's a little over 4. Reds, it's a little over 4. And Tegula are weird. And I just want to have, let's see, that wall, and maybe that wall. That's about 4 meter radius. So the footprint of a Picno hunting for 20 minutes on the bottom of the ocean is that big, right? If you multiply that by 700 million or 70 million, we got a lot of footprints happening on this landscape of fear, right? Every 20 minutes, if the Picno's active, of course. All right, so we wanted to know whether this is a temporary thing. They scare them for an hour and then they just come back, or does it stay? So this is the same data that I showed you a moment ago. This is the 1 hour mark. And this is again, relative grazer count with 0 being no effect. And you can see that for the first day, it looks essentially the same for all 3. And then by the second day, the abalone line has essentially disappeared. But the green line is still negative, it's still below the threshold, and it stays there for at least 3 days. And then for reds, it's doing something weird on day 2, but it's still suppressed on day 3. So I think we're seeing like at least a 1 to more like 3 or 4 day suppression of localized grazer density on an area this big, which is pretty cool. All right, so these are temporary things. These are like 1 day, 2 day, 3 day experiments. And we really want to know what's going on in the real world. And so we did a pilot last summer to see if we could just keep a Pycnopodia in a pen. And we're calling this the Pic Pen experiment. We have money to go back up to Sitka, and we're going to do 6 of these this year, half with Pycnos and half without for 2 months. And so we're putting these underwater. They're these giant pens, kind of like the size of the tables or so. And we're going to put a handful of Pycnos in each of those, stock at urchin densities, and track how many they eat, what their anti-predator behavior is, and hopefully we'll see if we get any kelp recruitment. We're going to also outplant kelp in there and look at grazing rates. So that's the plan for this year, and then ultimately, we have to put pigeon spikes on the edges of the things because they escape out of the thing. But this is all stuff in Alaska that we want to use to inform what's happening here in Oregon. So the Oregon Kelp Alliance is really involved in a lot of this. Aaron and I are both part of it. And so we just got funding from NOAA to repeat a bunch of these things here in Oregon, in Southern Oregon, probably next summer. And then hopefully do actual translocations and releases the summer after that. And so that big outplant is going to be called the Constellation. Are you ready? And I just wanted to say thanks to all the students that have helped. I have no idea how long I took.
Time for like 1 or 2 questions.
So in watching that video, it made me think, what have you looked at or thought about looking at the impact of having 2 of those sea stars present?
Yeah, we're gonna put 3 in each pen.
Okay, cool.
And then we have it all aligned, so it's like a chessboard. And we're gonna quantify like the Pycnos in A1 and all the prey are in C3 and C4. And anyways, we have a whole plan for that. Thank you though.
What do urchins eat when there's no kelp around?
Whatever they want. They are really, really good at eating all sorts of stuff. They'll eat anything dead, they'll eat anything alive, and they'll even eat rocks and plastic. So you have to watch your transect tapes because they chew right through them in like 5 minutes. Yeah, they will eat anything and they can live for like years without eating very little. That's amazing. Yeah.
Yes, do you know what the fear substance is?
No, I think it's just like a metabolic byproduct that they're queuing into. It would be really fun though. I love this question. So the Q itself, we think is waterborne, first of all, like it has to be emanating from it, right? But what I did last summer was a slime experiment and I took and I painted the bottoms of tanks with Picno slime. It wasn't very nice of the Picno, but it was only one. And then I released them and saw if they would like choose their sides. And I need to redo it because I think that they can smell slime that's even like, they can, if I only put it on the bottom, right? But I think they can smell it even if they're not right on top of it. And so I need to do some more replication and controls to figure out the like slime itself versus the collab, the pig pen slide, pig pen Q emanating from the slime. But yeah, they can smell it and I think it lasts like 3 days on the bottom because they were still scared 3 days later in the lab.
How about damage on specifics?
They don't seem to be nearly as scared of those. I did a bunch of the NSF reviewers said that they hated this, but I did a bunch of smashed up urchins and I stuck them in the tanks with the Picnos and looked at their fear responses. They don't really respond to them, but I was like calling that an otter mimic and they really hated that. So I might do it anyway, but that's not gonna be part of the new grant.
Awesome. Well, super
Yeah.
cool, Sarah. Thanks for sharing.
Yeah, no problem.
We have Matt from the Beretta Lab.
Okay, let's do the whole Zoom presentation song and dance here.
To go coach a baseball game. Sorry, I really want to stay longer.
Make sure you win.
Okay. They're 7, we don't
Okay.
keep smart.
Looks like we're good to go.
Sorry, team.
All right, okay, so thanks, Mark, for organizing this, I really appreciate it. So yeah, I'm Matt Powers, I'm in the Beretta Lab, and today I want to tell you guys a bit about the projects I've been working on to try to understand a bit more about the physiological and transcriptomic response of the intertidal copepod Tigriopus californicus to challenges of extreme hypoxia. So why do I spend all my days stressing out these poor little copepods with challenges of extreme hypoxia? To understand that, you got to understand a little bit about the complicated relationship that this animal has with its food. So their primary food source are photosynthetic microalgae. But these things also play a huge role in shaping the oxygen environment that this animal experiences in its splash pools. We put oxygen sensors out in pools full of T. californicus. We left them out there from July through September of 2022. What we discovered was that during the day, when the algae are photosynthesizing, oxygen levels are really high. But at night, when the algae switches to respiration, they consume all the oxygen, dropping it down to pretty much anoxic levels, close to 0 milligrams per liter oxygen. And that's well below the threshold that's considered severe hypoxia for most aquatic organisms at like 2 milligrams per liter oxygen. These events aren't just isolated, they happen over multiple days, consecutive days, back to back over weeks at a time. In fact, we had the sensors out there for 89 days, and of those days, 63 of them dropped below that threshold. On those days that went that low, they stayed that low for on average a little over 5 hours at a time. So these are really extreme hypoxia events that these copepods are experiencing. So one of the first projects that I worked on was to try to push the limits of what this animal can handle in the lab in terms of an extreme hypoxic event. So I thought, well, what's more extreme than just pure anoxia, 0 oxygen for an extended period? So I measured a couple phenotypes during and after anoxia. I measured survival, and then I measured another phenotype called loss of equilibrium, which is basically when an aquatic organism can no longer maintain its orientation in the water column. And it can't really swim around effectively either and go find food or mates. I did this test and I measured these phenotypes in a microplate respirometer. It's a closed system, just so I could monitor how long they were exposed to anoxia, know exactly how long I was exposing them. So this data took the form as loss of equilibrium, the proportion of individuals suffering from this effect over a 6-hour exposure to anoxia, which checks every hour, and then the proportion of surviving individuals after 6, 15, or 24 hours of anoxia. I also measured a pCrit in order to compare hypoxia tolerance in T. californicus to other species. It's basically just a statistic that lets you estimate how resilient an organism is to declining oxygen levels. It just represents the breakpoint at which an animal can no longer regulate its respiration rate. All you really need to keep in mind is that low pCrit values imply a greater tolerance to hypoxia. So I measured LOE, survival, and pCrit across 4 populations, and both sexes, but I only had the time to go over the population and species level stuff. 2 populations from Oregon and 2 from California, including our San Diego population, which is our genetic reference for the genomic studies that we do in our lab. So we'll go ahead and start by looking at the LOE data that I collected. Across the 6 hours of exposure to 0 oxygen, the 2 Oregon populations had a higher proportion of individuals suffering from LOE at every single time point compared to the 2 California populations. What was really cool though was that across all 4 populations, none of the replicates had 100% of the individuals that were suffering from this knockdown effect. Across the 3 time points that we measured survival, the Oregon populations again had a lower proportion of individuals surviving compared to the California populations. And it took until the full 24 hours of 0 oxygen for the 2 California populations, San Diego and Bird Rock, to even come close to the proportion of surviving individuals that the Oregon populations had. It was really interesting that even one of the replicates from Bird Rock in northern San Diego had 50% of the individuals still surviving after 24 hours of 0 oxygen. The pCrit values that we measured also replicated this difference between Oregon and California populations, where we had a lower distribution of pCrit values in the San Diego and Bird Rock populations. So looking at all these 3 together, it seems like we got some pretty good evidence of a latitudinal difference in hypoxia tolerance within the species. But how do our copepods stack up against other hypoxia superstars? Well, the species average in our data was just under 1 milligram per liter oxygen for pCrit, and the minimum was actually close to 0 in one of the replicates. And that replicate was in our San Diego population, which as a population had an average of 0.55 milligrams per liter oxygen. I did a deep dive on the literature and I organized a bunch of hypoxia tolerant organisms. And I made a little short list of the top 10 here, organized for you by descending pCrit values on the 4 right column. And T. californicus actually falls just behind these, including just behind 3 other crustaceans that are famous for occupying the oceanic oxygen minimum zones, another copepod species, a lophogastrid, and a species of krill. A really cool thing that popped out of this comparison was that the San Diego reference population actually ranked ahead of the goldfish, which is across the literature, it's actually referred to as one of the more hypoxia resistant animals on the planet in water systems. And I love that fact because it gives like a whole new meaning to Coach Lasso's advice. T. californicus had a lower pCrit than many deep sea species, which is really interesting. Species that inhabit depths of up to 1,000 meters or more. It's kind of wild that we can even make these comparisons because T. californicus lacks some of the key molecular components that most other animals use to respond to hypoxia. So for most other animals, when they deal with a low oxygen environment, they have hypoxia-inducible factor alpha and proline hydroxylases like EGLN to sense the oxygen and help them change the regulation of the genes they need to respond. The T. californicus is missing both those elements. And so one of the questions that we have in the lab is, well, what other components are they using to respond to hypoxia? And then one of the questions that I was interested in, which is how do those components respond to a rapid hypoxic event like they would experience overnight in their pools? So the second project that I worked on since joining the lab was to design an RNA-seq experiment where I measured gene expression over a time course of typical hypoxia exposure for this animal. And so I set up an experiment where I measured gene expression over 5 time points during the consumption of oxygen into anoxia and into a recovery period. So we have normoxia, which I'm labeling as control, Mild hypoxia around halfway through the consumption of oxygen. At peak crit, as determined by that San Diego population, an hour into anoxia and then 2 hours into recovery after those first 4 time points. What's really cool about designing a gene expression study like this is that you're not stuck with just doing pairwise comparisons between treatment groups. I could do pairwise comparisons between time points, but I can also model the effect of time as a continuous variable and look for significant genes that respond in like a linear, quadratic, or cubic fashion over these time points. And so I did that, and then I clustered the significant genes that came from that analysis and based on expression patterns. And so you can see here 6 expression patterns that came from our set of significant genes. It's some genes that went up over time, some that went down, some that responded more heavily in the recovery periods, like in cluster 2 and 5. And then some genes that clustered based on going up or down during hypoxia and then returning back to normal, like in clusters 3 and 4. And I can take those genes from those different clusters and I can pass them through a Go enrichment analysis, look for terms significantly associated with hypoxia resistance. I found lots of genes that work, genes that are known in other species to help with the switch to glycolysis, antioxidants, cell detoxification, and general stress response genes like your HSPs. But I won't just leave it out the generalities here. So I picked out a few interesting groups of genes that have to do with the unique biology of T. californicus. And so the first group that I'll go ahead and show you are some chitin and cuticle rearrangement genes. So what we saw is that most of the genes that have to do with the formation and pro-cuticle formation, pro-chitin formation were upregulated during hypoxia. So a couple examples like cuticle-like genes or OBSTEs. And genes that had to do with chitin degradation were downregulated during hypoxia. Genes like your deacetylases or your chitinases. And so we think that this has to do with the weird way that this animal respires. So they don't have gills, they don't have respiratory pigments, and they don't have a circulatory system. They just use passive diffusion of oxygen across their exoskeleton. So these changes just could reflect them trying to better hold on to oxygen in their tissues. The second group of genes I'll highlight have to do with the electron transport system, specifically complex 4. So we have a complex 4 assembly protein called COX-15 assembly protein. Helps with the biogenesis of this complex. And then we have hypoxia-inducible gene 1, which is actually a subunit of complex 4. And its job has been implicated in helping the cell deal with hypoxic events, increases cell viability. Both of those are upregulated at the anoxic stages and recovery stages. They're not really unique to T. californicus though, but I have another gene that's more unique to T. californicus and that most other animals on the planet don't utilize it. And that's AOX. Most metazoans don't have this gene. And this gene encodes an alternative oxidase, basically lets you have an alternative exit point from the inner mitochondrial membrane for electrons, and it stabilizes the membranes in there when you have lots of reactive oxygen species. And that's definitely the case when you have, you're suffering from anoxia and the reoxygenation when you're recovering. The last group that I'll highlight for you are pigment genes. So I didn't draw T. californicus bright orange just for fun. The photo shows that their tissues are absolutely loaded with carotenoids, specifically one type of carotenoid that is a really potent antioxidant. The third most significant gene in our data set was NINA-B. This gene encodes an enzyme that basically cleaves carotenoids and makes retinoids and antioxidants. It's highly upregulated during the recovery stages of the experiment. On the other hand, we've got 2 other genes, CD63 and SHRM2, and another species they're known to help sequester pigments into pigment cells or granules, and they were downregulated during recovery. So it could be that in the recovery stages, the animal is trying to no longer sequester carotenoids and is trying to shunt them towards antioxidant processes to help deal with reoxygenation. So I think that's probably all the time I have for the RNA-seq data, but I'll just leave you guys with a preview of what I'm working on now, which is a bulk segregant analysis using interpopulation hybrids. So the goal of this experiment is I'm basically going to phenotype hundreds of individuals, ideally a few thousand. I'm going to separate them into top and bottom performing bulks based on how they deal with hypoxia. And because the different populations of the species are so genetically diverged, it'll hopefully allow me to, when I sequence those bulks, map allele frequency differences between the high and low performers, and hopefully then associate that with loci that help with performance in the species. The phenotype that I'm gonna use is probably gonna be pCrit, just because that's a really high throughput method. I can just load up my microplate respirometers with copepods and measure hundreds of pCrit values every week, hopefully. So I'll go ahead and stop there just so I make sure I have time for questions, but just want to make sure that I thank Dr. Barretto for advising me and supporting me through his NSF grant, and make sure that I acknowledge my amazing lab mates who are really supportive, just generally wonderful and brilliant people. All right.
Awesome, thank you. Time for one or two questions again?
So this is fascinating, the difference between Oregon and California population. Can you talk more about why you think that is?
Sure. Yeah, we're speculating that it has to do with the coevolution with the algae. So down there, at that latitude, the sun is way more intense. It's out for longer during the year, and the algae really blooms heavily in the summertime. And so in those summer months with high UV intensity last longer in the south, we're thinking it could be that the algae down there blooms in the pools at a heavier degree. So those copepods have to deal with low oxygen events maybe more often throughout the year. That's just speculation though. We don't have oxygen sensor data from California yet, so.
And if you had something like Mid Coast, are you interested in looking at kind of like a gradient?
It would be cool to look at a gradient if we had the capacity to go up and down the coast. I just love driving, so I would just go drive up and down the coast and do that. Yeah, exactly. I don't know. I don't know if it would respond in an exact gradient fashion. I think it would have to be like a location specific thing. I think it'd be really highly dependent on the microenvironment where you're looking.
So because you see like a diol cycle of oxygen, have you looked at whether they maybe like front load some of these genes like in preparation for low oxygen events?
That's a good question. No, I didn't design the experiment that way, but that would be something to look at, is to see genes that are like maintained at high or low levels at all times. And maybe you could detect some of those based depending on like, if you're looking at different populations from different regimes, like maybe the ones that have to deal with it more often might have, differences in just baseline regulation.
Yeah.
Yeah, that'd be an interesting idea.
Awesome, well, thank you, Matt.
Thanks.
Right, next up, we have Dave from my lab talking about fly stuff.
Okay, most of you know me. Last time you saw me talk up here, or you saw me, I was talking about snakes and thermal biology. So I've jumped ship into the model organism system, and I'm digging it. It's fun. I'm here today to talk to you about Drosophila and looking at kind of the metabolomics of this accelerated aging phenotype we're seeing in populations that are selected for early reproduction. We're gonna get into kind of what that means in a second, but that's kind of, we're gonna take a small look at one piece of that story. There's a bunch of moving parts to it, but we're gonna get a little bit of it. So we'll hit on kind of the background of how this is set up. We'll talk about what we know about the trade-offs for this selection system, and then we'll go into what we actually found, or a bit of what we found within the metabolomics of it. So this is an experimental evolution setup. And for those of you that aren't familiar with that, you take an organism that has fast generation times, you give some of them some new selection pressure, and then have them have a bunch of generations under that pressure and compare them to each other and to where they came from, right? So we can see how they've evolved and adapted to this pressure. We can take that a step further and do what's called evolve and resequence studies, where we do the same setup, but we look at kind of the molecular differences between them, not just the general phenotypic differences. And traditionally that was done through mostly DNA work, but recently other molecular phenotypes have been incorporated, like transcriptomics, proteomics, and more recently metabolomics, which is what we're going to be looking at today. The main thing that we're looking at using this experimental evolution model in this case is going to be aging. And it's kind of a big phenotypic difference we're seeing, and we want to look at kind of the underpinnings of what's causing differences in aging between different organisms and their different things of doing. So we have to think about kind of the premise of what's going on with this and the foundation for how we ask these questions. And so we're looking at the natural selection, the course of natural selection, and how it plays a role with reproduction and kind of mortality rates or longevity, how long things live. And so the basic idea is that you have this point B here, where it's going to be for an organism the starting point where they first start to reproduce. And by kind of this theory holds that the force of natural selection is really high, or maximal before reproduction, because if you don't get to reproduce, you're no longer in the gene pool, right? So you have to have that really high level of selection at that point. And then once reproduction starts, that force of natural selection will slowly decrease over time until you stop reproducing, right? And the idea being how this works with age is that this reduction in selection pressure essentially works to, at that given age, an increase in mortality rate. So as that selection pressure decreases, your mortality rate for that same age is going to increase, you're going to be more likely to die. So that's the premise of it. So we use this idea to incorporate it into experimental evolution and kind of test it and see how things work, right? And so a really simple way of doing this is manipulating that first stage of reproduction. And that's kind of the selective pressure that we're adding to it. And so one way you can do that is you can delay the first reproductive stage, right? And what that'll do is shift everything to an older age, and that results in, from what we see or what we would expect to see, an increase in lifespan or increased longevity. You can flip that and make the earliest reproduction earlier, bump that faster, and that will result in decreasing longevity or having shorter lifespans. That's the idea that we would think would happen, we want to test. So this is the one we focused on. There's actually been fly models that look at all aspects of this, but we're going to talk just about the one where we are making reproduction happen earlier and how that's affecting things. So the basic setup for this, we're going to have, everything's going to be color coded. So the C types are going to be our control types. They're kind of on a relaxed selection regime. And they're going to be blue throughout the whole thing, and the other ones will be red. And so these guys they have a 48-hour window ending at day 28 for reproduction. So they have that 48 hours at the end of that for this 28-day generation cycle. Fairly relaxed as far as fruit flies go. There's not a strong pressure to it. The A types, or the accelerated aging types, have a much different regime where we've moved that reproductive window up to the 10-day mark, and they only have a 24-hour window to do this. And it's caused a whole suite of different effects and phenotypic changes, one of which is this increased development time, and then also a decrease in longevity, right? So it's a pretty big change associated with aging. There's been a lot of work on this already, and that we know some pretty big differences between these A and C type populations. And so there's been a bunch of studies looking at some major phenotypic differences between them. We also have some genomic and transcriptomic differences that are pretty strong between them. However, bridging those two things has been fairly difficult at this point. There hasn't been a lot of strong connections between the two. But the real short of it is that within A types compared to the Cs, we see they develop faster, but they die younger, they have decreased lifetime fecundity and decreased stress resistance. There's some pretty heavy trade-offs for this faster development. Recently, more work in our lab went to try and kind of help bridge this gap by using metabolomics, right? So that kind of highest level of that molecular hierarchy before you get to the full phenotype. And so they picked day 21 to compare between A and C types because we see some strong differences in phenotypes there, different mortality rates, developmental rates, things like that. So it was a good indicator of the differences between them. And they found some pretty strong differences. The real 1,000-foot summary is that there was some big differences in energy use and development, metabolites associated with those things. So that brings us to what we did here. So we're springboarding off of these ideas. And we really wanted to see, so those differences we see at this kind of static one time point, do those extend out across the lifespan? And are they representative of these other phenotypic differences we're seeing that we associate with different rates of aging? And so we wanted to explore that further using the metabolome. And thinking about how that changes over time, right? Using snapshots, it's hard to actually get a good picture of aging versus just differences in regimes. So back to these two different populations, we'll highlight here that because, as we talked about with the theory of aging, that reproductive window is going to be super important. So I just wanted you to see kind of where the comparison is on those with the stars or those reproductive windows. We got some different time points. So we have a sample from each that represents this pre-reproductive time point, which is pretty key because that's a central theme here. We have some early senescence time points, and then we have some late senescence time points. This is based primarily on mortality data as far as the senescence goes. You'll notice that the yellow ones are going to be overlapping time points. So there's 3 overlapping ones that we have for both samples, and then the other ones will be that earliest and latest ones are only for one sample. So we have 4 time points for each population, 3 overlapping. And the basic idea is that if the metabolome really is indicative of these larger phenotypes that we're seeing, that we should see some kind of difference between the selection regimes in the metabolome, not just representative of age, but representative of the actual selection regimes themselves. Because we know there should be differences in age as it goes across time. So we want to see how selection pressure changes that. We also think that there would be different trajectories of these kind of pathways over time. So here's the details of it, but we did a targeted metabolomic study on it with a total of 202 metabolites that were included in the actual results. So the first kind of results from this, we got a PCA for it, and just to orient you, day is by color here on the right, so their age, how old they are from A, and then the shapes indicate which selection regime they're under. And right away, we see that those overlapping days, where we have samples from both populations or selection regimes, they're different on both principal component axes. So we're seeing a huge difference between selection at that point, selection regime. Not necessarily a big difference between age, because they're kind of all underneath there. So that's kind of the first interesting note, that there's a big signal in the metabolome for selection. If we look over here at the day 9 ones from the A types, we see that they're very different from everybody else. So their metabolome looks pretty unique compared to the rest, and we'll come back to that towards the end. But also interestingly, we see that the day 70s, the oldest within the C types, the control populations, look more like the younger A types than they do the rest of the C types. So here we're getting a signal of different aging or age within the metabolome itself. So we've got kind of a mix of signals, right? We've got some developmental, we've got some selection-based and some aging-based signals. Yeah, so the question is, how does this kind of reflect aging? We see there's differences, but we need to look at the actual metabolites themselves to kind of understand what's going on there. So to do that, we did a linear mix effect model, looking at the impact of selection, age, and kind of the interaction between the two, and to see which metabolites stand out in these different groups. With the really basic idea that if selection is driving these phenotypic differences that we saw, we'll see metabolites being significantly different again for a selection regime, not just age, because we would assume that age is already going to be a thing that we're going to see a signal. And so when we did it, just to orient you here, the columns equal the significant metabolites, and then we have the different interaction terms, and so the orange circles indicate which ones they were significant for. So for example, we had 24 metabolites that were significant only for the interaction term. Does that make sense? We found a total of those 202, 174 of them were significant. So the majority of what we looked at actually was pretty strongly impacted by these things. Granted, this is a targeted analysis, so it makes sense that we would see a lot of them. But I thought this was cool that only 15 of those were unique for age. So we would expect there to be a strong signal with age, and most of that wasn't captured just by that age variable, meaning the rest of them have selection involved in some way. So we see there's a very strong impact on selection in the metabolome, which is a good signal for it. So we see that the signal's there, but when we look at the individual metabolites, we want to know how does the selection for early reproduction work with those kind of history trajectories, right? How are we seeing different aging based off of this? So thinking about these kind of life history trajectories, things we know so far from this study was that there was a signal of age and aging in the metabolome. I showed a little bit of that. There's some stuff I wasn't able to show that shows it a little stronger, but we have that signal within the metabolome. And that signal differs by selection regime. So there's lots of evidence I've shown for that at this point. We know that there's a difference there. But how does this actually look different in the metabolome over time? So we wanted to look at metabolome trajectories. So how within population, how their metabolomes change as they age, and then compare those between populations. We basically predicted that we'd see kind of one of two patterns within the metabolome. That we would see either shifted timing, which can look like an accelerated aging signal, where it's the same pattern, just shifted earlier, or a unique pattern, which we think of as like differently aging, where it's just a totally different pattern, they're doing something different with those things. When we think about the accelerated aging, other phenotypes that show this would be like mortality and developmental data, where we really just kind of see a shift to earlier ages in the A types. However, when we look at the reproductive data, like fecundity assays, they're different. They're not just a shift earlier, it's a totally different pattern that we're seeing. So to show this, there's a couple different ways we showed it. We're going to just look at the heat map here. So this is log 2 fold change between time points. So if we look here at the glucose, we're looking between day 9 and day 21, or age 9 and age 21, and the relative abundant or normalized abundance of metabolite for glucose between those. We see that it increased from day 9 to day 21 in that case, and that gives you the positive log 2 fold change value. So if it decreased at that step, you would get the blue negative values. So we can see some different patterns happening where we compare the A-type profile to the C-type profile as it changes over time. Oh, a couple notes. The asterisk indicates just a significance between that single step. So between, say, in this case, day 9 and day 21. However, when we looked at the linear mixed effect model, most of these were significant across, but this is just indicating for those very specific changes. And a quick note, this is abundance data, which can be really tricky to interpret. So if you have, well, just basically things are involved in many different pathways. So having a lot of it can mean different things depending on what the context is. So this is a pretty simplified thought process of how it's working. But we had those two major patterns that we were predicting to see. And we see that a lot of these amino acids are showing this kind of accelerated pattern, right? This trajectory that looks very similar to what we see in C types, but it's just happening sooner in A's. So that's one of the things of like the accelerated aging, a signal of that. And then a lot of the rest of these that we highlighted just are showing this different pattern, where it's not really that accelerated pattern, it's just something different is going on there. So I wanted to dig in further on that and kind of pick one thread that I thought was interesting to talk about. And it's looking at these carbohydrates, right? Because we have this really strong pattern that's centered around that day 9. We have really, really low abundances at day 9 compared to the rest. We don't see that type of pattern happening in the C types. So we're interpreting this as meaning they're using those carbohydrates a lot. They're burning through them at that day 9. So why? Why would they do that? So we go back to some previous work that was done looking at the fecundity data. And if you look at the A types on the top, we see that that day 10, which is their selection regime, they have a really, really high fecundity rate relative to the rest of their life. So their overall lifetime fecundity is decreased, but at that first day 10, they have this really high level where they're laying tons and tons of eggs. If you compare that to the C types during their reproductive window, which is stage 28, it's more than double that. So they're laying tons of eggs at this one window, and then the rest of their life, they're laying less. There's this kind of trade-off happening there. If we look back at that PCA information from earlier, the metabolome shows that at day 9, the A types look way, way different. So the day right before they lay the eggs, they look very, very different than everybody else. And if we remember back into the selection regime again, that day 9 is right before that window. They're larvae up until day 9, and then day 9 they become adults, and then day 10 is their last window for laying eggs. So they have this really short developmental time to be able to produce these eggs. And it turns out that we already know that at any given time when the fruit flies, when the females are ready to lay eggs, they have some drastic metabolic remodeling that goes on where they start using carbohydrates way more to produce eggs. And so it's kind of a known thing. So we think that's really what's going on here altogether, is it's just this crazy crank up of using carbohydrates to make this huge output of eggs very quickly. Then maybe there's some cost for it, that there's a trade-off later. So again, that optimization for early reproduction, we think it's altering their energy substrate use. And then because of that, that's part of what we're seeing is accelerated phenotype with amino acid use later that don't have time to show you, but looks like they start using that in place of carbohydrates later. Cool. So kind of the main conclusions from this is that the metabolome does have a signal of aging caused by the selection for early reproduction, right? The signals indicate we saw both examples of accelerated aging and of that differently aging kind of phenotype within there. And then we think that the selection for early reproduction is causing this metabolic remodeling to facilitate this rapid egg production, and that remodeling is potentially coming at a cost of like this glycolytic disruption later. It's causing this accelerated aging phenotype that we see in other ways. And for those two, we actually have some stable isotope tracing experiments planned to actually try and validate some of this and see where these different molecules are going. Yeah, this is such a short period of time. So that is a tiny sliver of what we've been doing there, but yeah. I don't know if there's still time for questions.
Yeah, we got time for that. Thank you. Yeah, we got time for one.
Yeah, so a question, it says, do you see any meaningful differences among populations of the same type? For example, AO versus ACO in terms of metabolic trajectories?
No. So we actually, as part of what we didn't show here, we looked at convergence between recently derived and long-standing populations under the same selection regimes. And they are completely converged at the mortality rate level, so that higher phenotype. And they're mostly, like essentially completely converged at the metabolite level as well. So they're really very similar under the same selection regime.
Good. Thanks, Nate. That was awesome. Sean from the Terry Lab.
Yeah, no more food on that. Okay. So, yeah, I'm happy to have this chance to talk a bit about my interest in combining paleo and modern data sets from particular terrestrial vertebrates. So animals today are dealing with climate disruption and lots of novel stressors in a variety of ways by moving around the landscape, changing their activity patterns, maybe changing their diets. And I think there are some fragments of information that we can learn from the fossil record that give useful context for these recent shifts. And I think that that would ultimately be nice to contribute to biodiversity conservation through that. So there's the idea of conservation paleobiology, which sounds very nice, but I think it has great challenges. And the biggest one, after seeing all the nice talks, is the issue that the paleo record has very low resolution. And so you have to try to think about questions that can be answered on both timescales. So yeah, I've always enjoyed looking at things on the ground and piecing things together. So I think that that's why I'm interested in that challenge. And in particular, looking at how animals have adjusted to water scarcity through time and across space. So I think that water scarcity is one of the most visible constraints for life. But the amount of water that's available is constantly in flux. And if you look at a picture like this, it maybe looks very desolate, timeless, like primordial earth or something. But the flux is visible here because of the mud cracks. You know, it's only through the expansion and contraction of clays that we get mud cracks. Ephemeral streams will leave washes. Glaciers might leave little scratch marks. And it's through then change through time on different scales, ranging from seasons, years to millennia, that we recognize what is a relatively dry or wet time. And so during my dissertation work on Madagascar, I was interested in looking at how animals responded to these changes over hundreds to thousands of years, because there were questions of what drove extinctions on the island. And recently, I've enjoyed thinking with Becca about how small mammals in the Great Basin responded to changes in aridity over the past 15,000 years. So I will not be presenting results in this talk at all. I was thinking about talking a bit about Madagascar, but I thought I would just talk about the ideas that guide what I've been studying and what I will be studying here. And maybe in the future, we can talk more about radiocarbon and stable isotope data. But yeah, I thought that I would start with this distinction that I think is possible to make in the fossil record between the habitat that an animal experiences and regional conditions at the time. And then talk about how certain modern studies can identify breaks from the fossil record, patterns in the fossil record, beyond just what animals are present, because there have been, of course, extinctions, extirpations, but also how the animals are responding to changes. So to introduce paleoclimate, I thought that I would start with this figure. So changes in regional aridity through time. I thought that it's helpful to separate these axes to conceptualize things in two different ways. One is with cartoons showing water availability on the left. So the lake is big and it's pretty wet. Then it gets smaller, it's getting drier. Move along the axis on the right, it might expand again. And this can happen over the scale of thousands of years or very short time scales, like we see in a lot of parts of the US. And if we want to extend what we can see to before we have historical records, then we have to look at fragmentary records like lake sediment records, these cores that we collected in Madagascar. And it's a big challenge to deal with that because you have to constrain the ages of the different layers and then look at proxies that are sensitive to different measures of interest, like water salinity, water depth, vegetation around the lake. So you rely on chemical analysis of the sediments themselves and macrofossils that are in there. And by looking at a series of records like this collected across the landscape, you can start to characterize these changes in regional conditions. So this is setting the scene for the animals, which we can consider, and their habitat aridity, which also varies through time. So again, we have the cartoon on the left. So in this case, during the wet times, the animal lives close to the water and its habitat is relatively mesy. As the water shrinks, this particular animal stays put and exploits whatever habitat's most abundant. So it starts to live in increasingly dry habitat. and it continues up. So the distance to the water is what is shown with the color codings of the drier habitat. And it might go back and forth along the axes to the right. And this seems really straightforward, but even just recognizing this pattern is exceptionally difficult in the material record, because you have to first characterize all of those regional conditions that I showed in the previous slide, but then also the responses of the animals. So you have to have a trace of the animals through time. You have to constrain them, anchor them in time so that you can match up these records. And then you have to have proxies to infer what is basically shown with the color coding here. So I think people could easily dedicate a career to any one of those steps, getting the material, dating it, analyzing it to infer something about behavior. I have done a lot of work with ancient bone samples to learn something about diet and habitat. But I think that one really cool line of inference is through poo and other biological materials that preserve. So these are rock hyraxes, and they have the cool quality of staying put over thousands of years and peeing and pooing around the same rock outcrops. And so their piles of urine cemented poo accumulate through time in these giant mounds. And if you look at any given layer, the chemical composition of the poo is sensitive in part to the plants that the animals were eating and how dry it was in the patches where those plants were growing. And so if you go through and get a bunch of measurements through time, then you can basically get a record of habitat aridity for these animals, which might show relatively dry or wet times. And the people who have done this research have then compared the rock hyrax poo records with records that are more closely tied to climate. So cave records, for example, where you don't have to deal with these intermediates of soil and plants, and animals. And what they find is a very close association between the regional climate records and the animal habitat aridity records. And so in this case, the rock hyrax is doing what I showed in the conceptual example earlier. It's tracking changes in regional aridity. And so this axis can be considered approximate. So yeah, I've dealt a little bit with poo, mostly modern poo samples, but most of what I've done is with bone. Bone has its own set of benefits and limitations, I think, of the fossil record. This is highlighting one of them. There's a lot more mixing with bones, and it's messy, but there are layers that accumulate, and sometimes those layers are preserved in caves like this in Eastern Oregon. This is a view of one of the study sites where we'll be working, called Paisley Caves. And so, yes, this is a pretty cool cave. It's looking out into a basin that was once filled with water. The water is actually what carved out this cave in the first place. And back then, there were a bunch of animals that tend to prefer wet habitat that lived around the cave. And so, yeah, this idea of preference, though, didn't come in with the rock hyraxes at all. And preference is a really big problem if you're trying to get paleoclimate information from biological material records, because it will introduce a systematic bias that will make it different than the climate record. So to illustrate, I just included another cartoon with a different animal that behaves a little bit differently. So it maybe is, for physiological reasons, more closely tied to water. And so when it gets drier, it sticks with its habitat. There's no change. And so in the figure on the right, there is a signal that's created then of this divergence. And maybe this divergence signal can be used through time to predict the extirpation of the animal in frame C if it's sensitive to water scarcity, and maybe its return in A. But yeah, this would be a really terrible animal to pick if you were interested in reconstructing paleoclimate. But it's pretty cool, I think, that there are these divergences and that they exist, because I think they're biologically meaningful. And the question that I have is whether a gradient through time in aridity like this can be used to predict where animals are living today across modern spatial gradients in aridity that exist around the Great Basin still, where we have arid basins and relatively wet peaks. So in other words, can we use this empirical relationship from the fossil record to learn something about where animals are living today? And in cases where that's true, then maybe the fossil record can be used to generate some expectations more broadly. In cases where it's not, and I can think of many reasons why there would be breaks from the fossil record, it raises questions of what has changed exactly. And I don't know exactly what to expect in this new study system for me, but I can give an example from my work in Madagascar where there's a break. And this is with my master's project where I studied two lemurs, an extinct one, Pachylemur insignis and Propithecus feroci, which is extant. These samples come from the Southwest, which is pretty dry, and regional records suggest that it was getting drier during the past 3,000 years. Despite that trend, though, these lemurs didn't have an increase in their habitat aridity based on the proxies that we were using. And despite ongoing drying trends, the Propithecus live in habitat that is wetter today than what their ancient ancestors used. So it's a bit of a break from the fossil record. And the question is, why would there be this break? And one answer that is pretty popular is that it's a bias in where parks are located in Madagascar. So parks protect forests that are close to water, and the surrounding landscape that's dry is occupied by people that are burning it to create charcoal and burning it to create land for livestock. And there are lots of dogs and cats and other introduced predators that live out there. And so really what we see today is the lemurs living in wildlife preserves. But the fossil record clarifies the observation that if lemurs were free to move around and they didn't have all of these other problems to deal with, they could probably exploit drier habitat than they do now in the face of climate change. So in this case, if these were lemurs, you might envision them breaking in this way, shifting down in the relatively arid regional conditions. And that's been called the ecological retreat on Madagascar by others. So we'll look for breaks, maybe not identical to that, but just breaks in general by comparing the fossil record with modern trapping data and fur data. We're giving animals haircuts and collecting data from the fur and the ancient bone of these animals that are kind of analogous. And so, yeah, I'd be happy to answer any questions about these different components. And thank you for your attention.
Great, thank you. Can talk to the reception. Okay, next we have Megan from the Berkeley.
Okay. So for those that don't know me, I'm Megan. I'm a postdoc in the Merck lab. So I'll be talking a little bit about the project I've been working on, which is the experimental evolution of antifungal drug resistance in Saccharomyces cerevisiae. So I'm taking us back to the realm of experimental evolution that Dave introduced, but this time using a yeast model system.
It's on the slide.
There we go. So one of the central goals in modern evolutionary biology is to understand the interaction between environment and genotype, or the genetic variation present in populations, and how these two things come together to influence variation in phenotypes, which ultimately can then influence fitness and adaptation of traits. So there's been a lot of work in studying this in the context of a simple environment with perhaps just one single environmental factor, and how that environmental factor may then impact genotype and phenotype. But there's been less work in understanding how this plays out in a more complex environment. So when we consider natural populations, they live in these highly complex environments where they may encounter selective pressures that can vary in space and time. And so as a result, these populations may be under periods where there's low selective pressure, as well as periods with higher selective pressure. And it's rare that they're only encountering one environmental factor at a time. They may have multiple environmental factors that can interact and that they're forced to sort of adapt to. And so this has sort of caused a push to understand how adaptation and dynamics of evolution play out in a complex environment. And so predictions actually suggest that this in turn can impact both the dynamics of phenotypic and genomic evolution. So my research is directly related to addressing this and empirically testing this in the lab to ask the question of how do the dynamics and evolutionary outcomes of adaptation differ in a complex and stressful environment. So to answer this question, I and others in the Berk lab, we use yeast as a model system for evolution studies. So particularly we use Baker's yeast, Saccharomyces cerevisiae. And so there's a lot of different reasons why this is a great model system, not just because you can make beer and bread out of it, though that's certainly at the top of the list there. But one of the things that I really wanted to highlight is that it can be cryopreserved. So we can basically freeze these populations at distinct stages of evolution, and then we can go back and revive those as needed to study them. And in particular, I work with an outcrossed population of yeast that we call 4S, and this was created by crossing 4 genetically and geographically distinct strains of yeast that I'm showing in the tree here. And so by using this base population, we have a population that starts with a lot of genetic diversity. And so this better models what occurs in natural populations that actually start with extensive standing genetic variation that can then be acted on. So using this population, I follow an experimental evolution approach where we can take our yeast model system, we can apply some sort of stress and then study the fitness changes that occur over time. And then also through the evolve and resequence approach, we can sequence the genomes of these populations and look for genetic responses that occur as a result of the stress. So this approach is really great to studying the effects of a lot of different stressors. My research in particular has focused on antifungal drugs as the focal stress. And so there's a lot of different reasons why I've been interested in this. Some of this is my own background and my PhD, where I was looking at antibiotic production by microbes. But related to this work, so there's a growing need to understand the evolution of drug resistance. If you've seen The Last of Us, you probably know zombie apocalypse is upon us, fungal infections are going to take over, but just kidding. It's probably not that serious, but still, more people are dying from these resistant infections. So there's been a push to understand sort of how resistance evolves and persists. It's also a very relevant system to study the responses to different strengths and combinations of selective pressures, which is part of my research. And so when we're developing these antifungals or antibiotics in general, one of the things we really consider is the dose. And then also recently, there's been a sort of a push to see if one way that we can tackle resistance is by making these drug cocktails where we combine multiple drugs and see if we can sort of overcome that resistance that we see to maybe just one drug. Also, the drug resistance phenotypes can be easily observed in the lab. So we can grow yeast in the presence or absence of a drug, and then we can track its growth rate over time by measuring cell density. So with this in mind, I want to talk about just one of the projects that I've been working on so far. And in this project, I evolved our yeast population in 2 different antifungal drugs as well as their combination. So one of the drugs is called caspofungin acetate, or CAS for short. And so this is a cell wall synthesis inhibitor in fungi and yeast, and it's been used to treat fungal infections that are caused by Candida and Aspergillus species. The other antifungal I've used is clotrimazole, or CLO for short. It is also a cell wall synthesis inhibitor, but its specific target within the cell wall or within the yeast cell is different from caspofungin. So this gives me the ability to compare across two different stressors, but still pretty closely related. So I evolved yeast in these two drugs by themselves, as well as a more complex environment that included the combination of the two. And then lastly, an environment that was just a control that received no drug. So I grew the populations in each of these conditions, and I would transfer cells daily to fresh selected media and continued that over the course of 14 days, which I estimate about 60 generations occurred during this time period. And so I would routinely throughout this experiment would archive these populations and then measure their growth throughout to see if we could actually detect changes of or evolved resistance over time. And so with that, I ended up with 48 total evolved populations or 12 replicate populations per 4 treatment groups. So I'll show just some of the results that I have so far. And one of the first things we want to look at is from a phenotypic standpoint, could we see the evolution of drug resistance in these populations? So to look at this, I measured the cell growth of our drug environments as well as our control populations at 3 specific time points. Day 1, which would be the start of evolution, day 7 is the middle, and then day 14 is the end of the evolution experiment. And so what we see, as you can see, the line goes up, particularly between day 1 and 7. It does seem that fitness was improving in these populations, particularly early on in the evolution experiment. It does look like they were able to grow better and more dense as time went along. And what was really interesting is when we look at sort of the degree of change that occurred in the different drug environments, those that were in the combined Cas-Clo environment, you see how steep that line is, particularly in day 1 and day 7. So this could be a sign that there's sort of changes that are different between those that were in the more complex environment versus the single. So that kind of gave us some insight into what's occurring from a phenotypic standpoint, but what about genetic changes that occurred? What sort of responses were we seeing at that level? So to look at that very briefly, it took these 48 total populations and I did pooled population genome sequencing at 2 time points, the start and end of our evolution experiment. And then look for genetic changes, particularly these single nucleotide polymorphisms or SNPs that occurred in those sequences. So after some filtering, ended up with 41,000 high quality SNPs that then became the focus of our analysis, which I then applied a linear model to identify those SNPs that changed most significantly over time. So using these SNP frequencies, one of the analyses I did was a principal component analysis. So here, each circle represents a replicate population that's color-coded according to the environment it was involved in. So there's a few things that really stood out from looking at this. One is that there's clearly differentiation among and between our populations. As you can see, some of these, even within a treatment group, really were spaced far apart. So a lot of genetic heterogeneity going on. The other thing that I noticed was that those that were from the combined drug populations, so these are the orange circles I'm showing here, they seemingly diverge from the single drug populations. So we see them cluster separately from those, which again gave us insight into the fact that there might be differences from those populations that were evolved in this more complex environment that we wouldn't have predicted from just the single drug counterparts. So one of the other things I wanted to do is get a better sense of what regions were changing or changed the most within these different populations. And so here I'm showing 4 different Manhattan plots where each circle represents a SNP. And on the x-axis is its position in the genome, and the y-axis is the p-value associated with that frequency change between day 1 and day 14. So the higher the SNP is on the y-axis, the more significant that change was. And so looking at some of the main findings we see so far is that many of the regions of the genome appear to be under selection. So it seems to be a very polygenic response, which is evident by the amount of peaks that we see popping out. These areas you see are considered a peak. So a lot of peaks in these plots. And when we compare across our different environments, we see that the SNPs with the highest significance of frequency change were actually from our single drug Cas populations. And that's evident if you just compare the y-axis between these different ones. You'll see that those in the Cas, that there were SNPs that were way more significant. And so from there, I wanted to sort of winnow in on regions that might be our top candidates that could be linked to this drug resistance phenotype that we were seeing. So far, I've just picked out 5 candidate regions from each treatment group. And it's a little bit hard to see, but they're kind of color coded here. And so if we were to stack these on top of each other, we could see that those top candidate regions in the combined drug populations were rarely shared with the single drug populations. They actually occurred in different regions. So again, this suggests that there's different changes occurring in the combination environment, suggesting that this could be like a non-additive response. It isn't simply just the effect of combining these two and having the same regions stand out. So there's still a lot that I have to do with this. I've just started to kind of dive into some of this work. But one thing that I wanted to look at is within these top candidate regions, what are some genes associated with those regions? See that slide. So on the left here, I'm showing a Venn diagram, and this is the number of genes that were in those top candidate regions and whether or not they were specific to populations in each of the environments. So we see in many cases, there's little overlap. There's a lot of genes that were unique to an environment. And then looking at some of the predicted functions of those genes, there was a lot of things that stood out, but I just want to highlight some that might be interesting. So there were some genes involved in cell growth, others involved in transport, and then some, which was most interesting, are actually known to be linked to drug resistance. In particular, PDR5 is one that really stood out as well. This is a gene that's been highly cited in the literature to confer resistance to one or more of the antifungals that I used. And so basically, I just want to sum up some of the work that I've just started to do, both related to this project and another project I'm working on. So in my future work, I want to look at how genetic trait architectures change in response to different strengths of selection. So that's actually the evolution experiment that I just started this week. So my first experiment, we're looking at just one drug dose. This next one, I want to consider what happens when we look at multiple drug doses. Also want to look at the question of how repeatable or parallel these responses are within and across my populations, and does that change depending on the complexity of the environment or the strength of selection. And then lastly, hope to dive a little bit deeper into what these changes might be driving at the functional level and look at how patterns of gene expression or chromatin accessibility change in response to these different environmental contexts. And yeah, so just to sum up and thank everybody in the Burke Lab that I get to work with, especially my mentor, Molly, as well as the undergrad assistants that help with this. And then special thanks to Mark, who a lot of his work in the Burke Lab as a postdoc kind of laid the groundwork for what I do now.
Awesome. Well, thank you, Megan.
Yeah, very cool. I wonder if maybe you've already done this, but I wonder if there's any sense in testing because you found no overlap in the peaks somewhat, you know,
Yeah.
very little overlap.
Yeah, very little.
Like getting the one that evolved in the cocktail and testing whether it evolved resistance to individual drugs. I mean, like if they're using different combinations of genes, did it completely escape, you know, the ability to evolve to be resistant to individual drugs or not, because it's almost like this, the switch could be driving them in a different direction, but when you combine them as a third direction,
Yeah,
we have the ability to probably kill them with a single drug later on. I mean,
yeah, it's a really good question. I think it's something that we're kind of considering too, is doing more work on the phenotypic end as well with these populations and looking, taking these evolved populations, going back and screening them either in single drugs, both that we included as well as others too.
I don't know,
Yeah.
Mike?
I just had a comprehension question. Can you go back to the slide where you just showed the phenotypic change in the very beginning?
Yeah.
And they got very resistant. Yeah.
Yeah.
What's on the y-axis?
Oh, so this is basically I'm showing the relative density compared to the control. So essentially we would consider at each of these time points the control like at like 100%. And so it's how much these test or treated populations grew relative to that. So just simple like division.
So day one is they've been treated for a day.
Yeah, day one is essentially 24 hours in the selected media.
So like that one on the right. So relative to the control.
Yeah, so that's, if that's what's throwing you off, so that one does look a little funky that it's at 0, is that what's confusing you here?
Well, just that they're all at different starting points.
Yeah, so I actually could not measure the OD of the combined drug population after 24 hours because it grew so little. I actually had to wait for an additional 24 hours before I could
Oh, I see.
transfer those, yeah.
I see. So this is after a day of treatment, and like on the left, they've dropped down to like 30-some% of the controls just after one.
Yeah.
day.
Yeah.
I'm also curious, what do you make of all those peaks in the control at wow.
Yeah, so we know, actually kind of related to some of the work Mark did as well, that there's just general laboratory handling responses that we see by just basically growing these in the lab, routinely transferring them. So we do see some sort of selection signatures that are just unique to the laboratory handling process. So we always include the control for that reason.
Okay, awesome. Thank you, Megan.
Great. So hi, I'm Maria. I'm relatively new. I started in January in Virginia Weiss's lab. So today I'll be talking a little bit about my dissertation work and how that ties into what I'm currently working on in the lab. Which is all around partner specificity in symbiotic cnidarians. So cnidarians are things like corals, sea anemones, jellyfish, and many cnidarian species form a symbiotic relationship with single-cell microscopic algae, where the hosts provide shelter and inorganic nutrients to the symbiont in return for photosynthetic products, which it can use for energy. So this symbiosis is really important because it forms the foundation for coral reef ecosystems. And part of its success comes from this symbiosis because these habitats are relatively nutrient poor, but it also makes them vulnerable to stress. So this is actually a bleaching event happening right now in the Great Barrier Reef, just emphasizing how important it is to try to understand this symbiosis and how to make it more resilient. So we know that there's variation in resistance and resilience to stress in coral, and some of it is due to its algal partners. So the algal symbionts are an extremely diverse group. So they associate with 5 different genera. So it's very diverse family of Symbiodinaceae, it's called. And with that comes a lot of functional diversity. So these are just some examples, right? If coral associates with one symbiont species, it might give more carbon to the host and grow more, or different symbiont types can also be better in some environments than others. But there are limitations, right? Not all are compatible with each other. And we have a very basic understanding of why this is, why we see these associations in some conditions, and why there are these limitations. So I'm interested in the drivers and mechanisms of partner choice in this system. So when I say drivers, and I'm not going to go into all of these, but I'm thinking about coevolution, ecoevolutionary dynamics, and then the mechanism. So what's actually going on at the cellular level. And I just listed some things here that we know is involved in healthy symbiosis, but we don't know how it's involved in symbiont communities or that association, and if there is any sort of choice that can happen. So in the Weiss lab, we use the small sea anemone, Aptasia, as a model for coral symbiosis. So this anemone associates with the same species that coral does, but we can make it totally aposymbiotic. So decimated symbiont populations, infect it with different symbionts, and control for its symbiont community. Also, it reproduces both asexually and sexually. So the advantage here is we can leverage asexual reproduction to control for host genetic background as well, and generate new genetic diversity, naive larvae that we can then also infect with different symbionts. So during my dissertation, one of my chapters focused on looking at the genetic and environmental effects on symbiont communities in natural populations. So as we heard just now about how labs can kind of select for certain things, most studies on this system say these anemones are very specific to their symbionts. But there haven't been many that have gone out into the environment and seen what they associate with. So I sampled Aptasia from 6 different populations in Florida, spanning an environmental gradient, and sequenced the host and symbiont. So first we'll take a look at the symbiont communities. So first, it's super diverse, super colorful, right? So here each panel is a different population ordered from north to south. Each bar is an individual, and then the colors are the symbiont species. So each species is a color. The gradient within that is variation within species. So it's a lot, but what I'm trying to show here is there does seem to be some environmental component. As you go further south, it's mainly the blue symbionts, and you kind of see a loss of the red, orange, and purple types. And this could be due to their tolerance to potentially temperature or differences in light that you see along latitudinal gradients. But you can't totally rule out genetics, right, from looking at this. This doesn't show anything about relatedness. So maybe, right, the blue that you see up here are more related to these ones, and the orange, the hosts that have orange symbionts are more related to each other. So that's what I looked at next. So here on the top, these are clustering of the host, and then on the y-axis is clustering of the symbionts. So if there was this sort of co-divergence, co-evolution happening, you would expect to see kind of like chunks of those colors, so more closely related hosts having more closely related symbionts. No, that is not what we see. And in fact, we see that they can also associate with many different symbiont genera. So each chunk there is a different genus of symbiont. So it's very diverse, and that seems to be shared across these different host lineages. And to further drive the point home that no, it's probably not the host genetics, they do reproduce asexually, and we were able to capture clonal lines. So each grouping here is now a clonal line. The columns are an individual clone. And so you can see they have divergent symbiont communities, and that's not what we see in the lab. We have lines and they all have the same symbiont, but potentially we are selecting for the symbiont that does best in the lab I don't know. That's why I'm here. So, just to summarize this, so really, we don't see much of a genetic component, maybe an environmental component based on biogeography, but that's definitely something that needs to be validated and looked at in the lab. But I also don't have any environmental data. So everything I've shown is what's inside of an anemone. It's not what's out there in the environment. So there still might be some sort of selection for the symbionts it's associating with, even though it's extremely diverse. So this is work that I've done over the past couple of months. It's very hard to see these larvae on the screen, but we've been getting aposymbiotic larvae in the lab. And so I infected them either with a maternal, the maternal symbiont type. So I offered them the maternal symbiont type or a different species. And so this species I didn't really see in my data set much, but it associates with a jellyfish that co-occurs with these anemones. And I know because it stung me a lot when I was sampling them. So I offered them these two types, and then I looked at symbiont density over time. So the x-axis is days post-infection, so 3 to 7 days. And the first thing I just looked at was, are they getting infected? How many symbionts are there? So the maternal type looks like it infected pretty well, and it also divided, the population increased over time. Whereas the other type didn't infect very well and actually decreased a little bit over time. And so it's not like, wow, it's totally surprising. It's just incompatible, right? But what I found really interesting was the ones with the jellyfish symbiont actually survived longer than the ones with the compatible symbiont. So I went back and I measured the larval size, so the size of the host. And so the reason why I have the aposymbiotic one, no symbionts here, is because you can see that their size decrease over time. They're using up their lipid stores, getting smaller. And you see the same thing in the symbiotic one. So maybe there's a slight, it's giving some energy to the host, but it's not enough to really compensate. Whereas this other jellyfish symbiont, I think it's just eating it. So it maintained size and it survived longer, and you can see, so these like not filled in points are no symbionts. So over time, there's less symbionts, they stay the same size, they're probably just eating it. And this is actually new, we don't know this, so we don't know how that happens in corals or anemones. There's one paper about symbiont selection, and it shows this bamocytosis pathway. So this is depicting how a pathogen would infect a host. We think for a symbiosis, it stops about here. So it ingests the symbiont and then it stops. The symbiont doesn't get killed, it doesn't get expelled. But somebody, another paper offered non-symbionts and they just spat them out. So bamocytosis. And that's the one paper that we have on symbiont selection. I'm pretty convinced that they're digesting these symbionts and digesting the jellyfish symbionts. And this is, I think, more interesting because we know it's a symbiont. We know it associates with jellyfish and coral. But for some reason, the host is eating that one, keeping the one that is compatible, but is actually hurting its fitness, right? They're actually surviving less, costing the host compared to the jellyfish symbiont. So I'm really excited to dig into what the mechanism is of this. So stay tuned on that, just getting started. So I just want to acknowledge my collaborators and advisors. So Sam actually helped me with my dissertation work, if you're familiar with him, and that's how I got here essentially, and my funding sources as well. And I'll take any questions.
Well, people can talk to you later. So lastly, we have Justin from Ben's Lab.
Lastly, indeed, I'm aware that there's food right to that door, so I'll be respectful of your time. Hi, I'm Justin Priest. I recognize some of you. I'm an FRA working with Ben Dial, who has an appointment in IB, but also over in mathematics. And I work with him on projects centered on epidemiology. Currently, he's involved in evolutionary ecology, but he also has an appointment over in mathematics. So I worked over in botany and plant pathology for about a decade, helping with comparative plant genomics work. And now I've been over in IB for about 4 years, starting on the TRACE project, if you recall that fondly or not. And stuck with Ben after that to continue doing some work. My talk is pretty different from everything you've heard today, which is pretty cool, actually. So you've seen a lot of great experimental work. I'm currently working on an NSF grant that was given for prototype development. And so the first part of this is just going to be sort of a tech backgrounder. It's more of a tech talk. And then the backside is about how we're doing some modeling using that technology as we move towards enabling future experimental work. Okay, so just to lay that out. Graph databases. Who in here works with any sort of data that they would consider characterized as network data that's connected like nodes and they have relationships of a few? What, if I, not to put you on the spot, what platforms do you use to characterize that network nature of that data? Like how do you analyze the network? Load it up in R, run scripts, GraphX, NetworkX, stuff like that.
as well.
Say it again.
CQSL.
CQSL, right, okay, cool. So I'm going to talk about the development of graph database platforms as another way to take a look at characterizing networks, especially very heterogeneous networks of very large data. They're useful for visualizing that data and also useful for doing analysis, especially like at the code level. So a quick background, I'll gloss over this and get to the project I'm working on. Graph databases, you're probably aware of the basic concepts of graph. You've got nodes, you've got edges. The edges connect the nodes. They may have directionality to them or they may not. If they are directed, that could indicate that one node owns the other one or is aware of it, belongs to some class of thing, can reach something else. So, you know, it performs a process action on another one of those nodes. Graph databases are basically structures set up to represent that kind of data. I hope to show you a few different ways that this might be relevant to some of your work. I've already seen several experiments that this might be useful for. So just as a quick, like a facile example, we've got a couple people, the people represent nodes, Alice and Bob. There's some properties associated with them. We have their ages and an ID, and you see that they're both connected to another node that is a group. It's chess, the chess group. So they're called the chess enthusiasts. The relationship is membership. Alice is a member of the chess group. Bob is a member of the chess group, and they know each other, right? So when you characterize data like this, you can end up gaining efficiencies over very large scale sets of data because you don't have to list things redundantly, for example. So if you work with tabular data a lot, if you are a CSV gunslinger and like you load up massive files into R, like this sort of thing might be useful to you because it's can lead to a lot of computational efficiency. You may be well aware of graph theory in general because of genome assembly, right? Michael Waterman, I believe he's the keynote speaker for commencement this year. It's totally cool. Yeah. So one of the main creators of our entire theoretical structure about how to assemble genomes is involved in graph theory. Some other ways to think about networks and how they're used in the spaces. I saw Go enrichment analysis earlier, right? Okay, so the good old gene ontology, ways to structure knowledge. So ontological definitions that could represent things like superclasses, really generalized things at the top of a network or a tree, if you will, and then far more specific things further down. So that leads itself to all sorts of interesting like network math behavior. You can do traversal along the network, you can follow paths, you can do pruning, you can do branching, that sort of stuff when data is characterized like this. With Go specifically, you can describe processes. So these can be nodes. The meiotic cell cycle can be a node. You can have activation reciprocal meiotic recombination, that the relationship is that node positively regulates the cell cycle, for example. So they have lots of different interesting features. I'm going to talk a little bit about one particular network or graph database platform just because it's the one I've been using most recently. I don't work for Neo4j, but I found it pretty useful. So it's a platform that's been around for 6 to 8 years and has gotten really, really popular. It's heavily utilized over in the private sector too for business analytics and social media and things like that. But at its heart, it is a network descriptor database, and it has a visual interface, but it also allows for all sorts of great computational access. So if you can code in Python, for example, there's a great Python interface for this that you can use. When you characterize data like this, we may be familiar with this sort of thing. It's nodes and edges in this particular case. I think we're describing, yes, pathways. Some other folks were talking about their metabolic pathway analysis, for example. And the data is still in there, right? So if you want to get it out, it can come out as JSON records. You can identify this node is a pathway. It's got all these attributes associated with it. So each one of those little circles on the graph behind there, if that's a node, in addition to its connectedness to all these other nodes, it can just house its own properties. That's where it becomes really powerful for things like population modeling, because you can store demo information alongside your node, and then you can show the connectivity between this starfish and that starfish. Just on the tech side of things, if you've ever worked with a relational database like SQL, the value of a graph database really has to do with computational efficiency. So if your data is tabular and has a lot of holes in it or a lot of null values, those null values take up space in databases. And when we're talking about millions and billions of records and millions and billions of relationships getting up into that terabyte, petabyte kind of space, then that can be really, really inefficient on the storage side. It can also be very inefficient on the computational side because most of that data is not pre-composed. So you're going to have to write the kinds of queries that hoover up large sets of tabular data and then infer relationships. Graph databases already have the relationships because that's how the data is encoded, right? And it doesn't store the null values because it doesn't need to tell you what isn't there. Now I understand there are obviously circumstances where you do want to know that, but here's a great example. Like it's just some table data I pulled from somewhere. ID, it's all different, but it's just redundantly storing the same information. A lot of rises to time over and over again. That sort of thing doesn't show up in a graph database because there's really just one representation of a rhizus sativa as a species node. All sorts of stuff is connected to it in different ways. I won't get into the tech details on this, but there are query languages that are specific to graph databases. Neo4j has one called Cypher. So if you're used to like writing queries in SQL to extract data to work with it when you're coding or like directly on an interface, there's a similar way to do it here, but your brain works in a completely different way. When I got going with network stuff, I had to completely rethink, I'm not really dealing with records anymore. And I'm not really worried about having to like create generic relationships to connect different types of records together. That work's been done and or I can ignore it. And instead I'm looking for types of things and types of relationships that connect things, just as a conceptual reorientation. Just some example projects. In the past, I worked on something called the plant reactome. It's a derivative of the human reactome project. So those of you that work with anything having to do with cellular process, metabolic, developmental, otherwise, you can describe cellular process cycles as networks, right? There's the membrane, there's stuff on the membrane. Those are all nodes. And then the lines drawing in between them are like process directions that upregulates that, downregulates that. You can characterize cellular activity as a network. Just another way to think about using it. You can categorize hierarchies of pathways as a network. Okay, to the project I'm currently working on. Pandemic resilient cities. NSF has funded us to do some developmental prototyping. The goal is to help cities, urban environments, find ways to be more predictive and more resilient to future novel pandemics, things that we don't know that are coming. In order to do that kind of work, we need to be able to try to predict patterns in disease transmission, especially patterns in novel disease transmission. So we're partnering with a few different communities in Oregon. We've started already surveilling with Bend. And Our particular modality focuses on 3 different areas. We've got the mathematical and computational modeling. That's where I come in. That's where Ben does a lot of his work. We've got engineering and wastewater surveillance folks. A lot of the folks I'm working with are like, we all worked on the TRACE project together, so you'll see common threads here. And then a major component in public health and community engagement. As we all learned from the last pandemic, if that piece isn't in place, it's really difficult to help communities fight off novel disease. So how's it work? Basically, we're working on developing prototypes of sensor systems in cities. Wastewater surveillance is a reasonable example, but it's not the only one, where we lay down sensors in the wastewater grid in urban environment, and we pull the data for that. We're looking for right now like non-novel diseases, but you know, we're pulling COVID, flu, A and B, RSV. We've talked about doing some others, we haven't yet. And what we're looking for is patterns that we can model on and then use that as predictions for the future. How does disease move through a community? And then how can we use that information to help the community avoid getting as much disease in the first place? The middle piece, digital twin, has to do with forecasting. So that's where Ben and I come in. He's doing a lot of modeling and forecasting work, and I'm helping build the models on the technical side of things. Briefly on the modeling. So another example of the network, which is why I've been talking about graph databases. Wastewater surveillance. On the left-hand side, we just wanted to show how wastewater was flowing through a system. That's directional. Okay, it's moving from smaller catchment points through larger pipes to larger catchment points to treatment plant. And we wanted to show how that was connected to places, synthetic places, putative places. Okay, we're not talking about specific people's houses or specific people, just to be clear. So in this case, blue dots are maintenance holes where we're pulling wastewater surveillance. Red dots are places, businesses, homes, etc. And green dots are people. And we're able to construct a network that lives in a database like Neo4j that includes directionality, and we can use to infer about relationships. So if you detect disease at one particular maintenance hole, wastewater surveillance point, then you can infer from that that the transmission might be localized to this area, for example. If the big picture here for epidemiologists is that if you want to reduce transmission or prevent transmission in the community, you want to drive changes in infectious transmission at population level by disrupting contacts, so you don't have as much hazard, right? So on the right-hand side is a high hazard situation because of these connections between these different groupings on the network. The blue area could represent infection, for example. And then on the left-hand side, if we remove some of that connectivity, then you have less transmission and less infection. And that's just represented down here by here's your R naught and here's your relative aggregate measure of contact hazard. We want to be in the green zone, not in the red zone. These connections are not necessarily person to person. They can be behavioral in a community. And this is where we're trying to help places. So like, for example, it could have to do with a high aggregation of shopping activity in evening windows in a particular area in a town where there's a shopping center, okay? Not that specific shopping center, but that type of behavior, right? So then if we can help cities to advise them on policies that don't like necessarily target people and shut everything down and make them stop doing what they're doing, but rather look at more aggregate behavior, we might be able to control for some of the disease transmission in ways that actually work for communities. So Python, yada, yada, I code stuff up, I pull data out. Like I work from Ben built some synthetic populations for our digital twins. So the last slide here is that we can do wastewater surveillance, and that could be a representation of networks. But another way to represent networks is like a synthetic population. So to my title, population modeling. Again, we don't want to target specific cities and specific people or specific demographics. So we're working on developing synthetic population models that carry behavioral characteristics of large cities. So Ben has been developing randomized sets of populations. Like I'm working with a small one here recently, about 10,000 people, about 3,000 places, and then randomly assign connectivity between those fictitious people and the places they frequent, their homes, their places of work, et cetera. Loaded that up using Python into Neo4j, into a graph database, I get a network, right? Initially, the network is showing, I switched the colors on you. Blue is people, green is places, There's no maintenance hole, no wastewater nodes in this system, it's just people in places, okay? And then the red, see how some of the lines are thicker? We created relative strength of contact between different people and places. So like this person X might have like first order contact with this place because they go to work there every day. Second order because they like drop kids off at school here, on down through 10th order. So there's relative weakness and strength of their contact with a place. So we built this, but now here's where it gets interesting with network theory. You can then create and add relationships inferred on the basis of what's going on in this population, synthetic population. So what I did was I took pairwise connections between person A and person B sharing a common place. And I made note of the strength of their relationship to that place. And then I created a new relationship between those two people. And on that relationship, I calculated it's an inverse product of just their contact hazard between those two people concerning that place. This thickens the network, okay? So we just had lines initially showing singular relationships, and now we've got it between a bunch of people, right? And you may remember on the previous slide, I just showed how we want to try to lower contact hazard by severing behaviors or connections between people and places. Well, I experimented with that idea with the synthetic population. On the lower portion here, you see it's really dense, lots of people connecting in lots of places, and the red thick lines are like the highest order of contact, like where people live together, coexist together. That's what I then looked at like what was one of the highest incidents of places where everybody was meeting. What happens if I cut that place out of the network? By making, like encouraging via community policy people to not engage and go into that place, for example. Well, this is the result, right? So I pulled that place out, and all of a sudden I had much less interconnection between all the various people and the remaining places. So that's sort of just proof of concept showing that we can thin down contact and thin down transmission in spaces. Calculated contact hazard from there. And next steps from here are math that I rely on Ben for heavily. And he's great. He's given me lots of good directions to go with aggregating this data and sliding windows, all kinds of fun stuff. Of course, you always have to be careful. You could just end up with another hairball. We've all seen those in network land. Even with just 13,000 fake people or 10,000 fake people and 3,000 fake places, I still have 1.6 million connections after just a couple of relationships. So high performance compute is important. And our next step is along those lines. Ben just bought a really big beast of a GPU machine, a Grace Hopper that's going to go in the new CIC. And so we're going to be able to like really scale up and work at the level of actual city size with our synthetic or digital twins for those cities. Hopefully, we can create some populations in the synthetic space that help inform and drive policy so we can keep people from getting sick. That's ultimately the goal of the work. I thank Ben and all the other folks I'm working with. I don't see many of you around here because I've worked almost exclusively on campus. Like all these other folks work on campus. So we have our meetings there when not online. And I didn't want to take a whole slide for it, but we also do cool outreach stuff like we had a science fiction writing contest as part of this project and got a bunch of manuscript submissions and had a celebrity judge and selected winners and they get t-shirts and money and it was a lot of fun. So the 45th Parallel, the on-campus lit mag, they're publishing those at the beginning of June. So if you're into that, check them out. There were 4 very good sci-fi short stories were selected. So anyway, thank you for your time and your attention and for being patient. There is food. I will answer questions, or I'd be happy to take them over in food land. That's okay too.
Awesome, thank you. Any questions? Okay, all right, talk later. Yeah, thanks for coming out, everyone. Thanks, a lot of fun. And yeah, we have some food and drinks